right, let's go ahead and get started. Good afternoon, everyone. Hope you had a great weekend. Just a few announcements. So almost all of you submitted the pledge. Almost all of you did it on time. There is a small fraction that did not submit it at all, which makes me very concerned about, so if that is you, please, you still have a chance to submit it. I will still read it. Some of you submitted it and it was blank or not the full thing. I've sent you a message and given you the opportunity to send it again. So please pay attention to that. They have all been graded. You can look and see. If you got a 1, great. You've done it. You've completed this. No problem. Many of you also took advantage of submitting your homework for extra credit. So many of you did it by Friday at 10 a.m., and so you get 1 full point of extra credit for doing that. And then many of you also submitted it by noon today, and so you also earned a half point for that. And at noon, you probably noticed I then posted an answer key. So if you already went to the discussion section, you've gone over the answers, you know it. If you feel the need, you need to double check something, you can look at the posted key. But after noon on Tuesday, you don't need to submit the homework anymore because you're not going to get any credit for it. But certainly still do it, still go over it, make sure you went to discussion section and you're clear on that. Any questions? All right. Oh, no. Did the internet go off? All right. So it's a really exciting week for science, right? Why are these 3 people smiling? They won the Nobel Prize for what? Immunology was the topic, and so yesterday they announced the Nobel Prize for Physiology or Medicine, and indeed, these 3 scientists, one of them is an alumnus from UCSD, so he actually got his bachelor's here, which set him off on course to do great science. And what about immunology? What did they specifically kind of discover? Regulatory T cells, yeah, there's a little hint down here. And so the Nobel Committee announced that they were awarded this for a fundamental discovery about the principle that keeps our immune system in check. Our immune system's super important, right? It defends us against foreign bacteria and viruses and things that can really cause damage to us. But we also don't want our immune system to get activated when it shouldn't be. This can lead to autoimmune diseases. And so that's essentially what they helped discover, is that our immune system has these regulatory T cells that keep our immune system in check. And so unfortunately in this class we're not going to have time to talk really about any immunology, but many of you are probably interested and will go on and learn all the intricacies of immunology, which is at its core molecular biology, right? If you're interested, I have also posted the Nobel Prize Popular Science little article about it in a module on our class website. Again, you won't be ever tested on that, but it's a nice description of what these scientists did and how they discovered these regulatory T cells that are able to calm down our T cells if they happen to attach to a cell that has a normal protein in it and should not be activating our immune response. All right, so last week we spent the Tuesday first talking about the major types of DNA sequences that are in our genome. And then on Thursday we learned about how all this DNA sequence, over 3 billion base pairs of DNA, gets compacted to fit inside each one of our cells. And so we learned that our DNA wraps around a core of 8 histone proteins. That complex of 8 histone proteins we call a nucleosome. And then these nucleosomes can further compact. And when you have chromatin that is very highly condensed, very highly packed together, we did talk about a particular type of histone modification that helps that chromatin be in a very compact state. And which type of modification was that? Methylation, right. So especially on histone 3, lysine 9, you can have mono, di, or trimethylation, and that helps keep the positive charge, and it helps the chromatin stay compact. On the other hand, if we have genes that need to be expressed, that need to be transcribed, we want our chromatin to be a more open, a looser sort of conformation. And what general type of modification is usually associated with open chromatin? Acetylation. And in fact, we talked about that same position on histone 3, position 9 lysine. If it's acetylated, that's more associated with open chromatin or active chromatin. And then we talked about a type of experiment we could do to actually test whether in a particular cell type or under a certain condition, a certain sequence of DNA, a certain gene, is indeed in a more open or a more compact chromatin conformation. And what is the name of this experiment that we talked about in class last week? A DNase protection assay. We are asking, is our gene, is our DNA protected because it is tightly wound up in condensed chromatin, or is it loose and open and therefore can be chewed up by DNase? So if I were to do a DNase 1 protection assay and I wanted to test what the chromatin structure was in 2 different cell types, what's the first step I would do? I want to isolate the nuclei of those 2 different cell types, right? That's where the chromatin is. So you isolate the nuclei, you have those in your 2 different test tubes, and then what are you going to add to each of those test tubes? DNase, right? This is the whole thing. You're asking, is it protected from my DNA? So you put the DNase in the test tube. It will go around and try to cleave up any regions where it sees vulnerable DNA. If the DNA is all wrapped up, it's not going to be able to get in there. After you've done the DNase treatment, what is the next step? So you're going to want to do a restriction enzyme digest, but is that going to work if you still have all this protein in there? No, you need to purify the DNA. So after you do the DNase, then you want to get rid of all the protein, everything else, and so you're going to have purified DNA, and then you choose a restriction enzyme that will just cut out a fragment of DNA that can run into a gel for your southern blot experiment next. So restriction enzymes are these enzymes that recognize specific sequences of DNA and will cut. Once we have our cut DNA, as I just said, we're going to do a southern blot experiment. And so the first step of that experiment is to take your DNA and do what? You run it through a gel, right? You use gel electrophoresis, which will separate those DNA bands based on size. The gel's kind of flimsy, so you want to transfer it to something more stable like nitrocellulose paper. Once it's transferred to there, you fix it, which just makes sure that the DNA stays in that same pattern on your nitrocellulose. And then what are you going to use? You're going to use a probe. You're going to use a probe to ask, is the specific sequence I'm interested in this case the globin gene, is it available? Was it protected? Or did it all get chewed up and I'm going to see nothing at the end there? What are the 2 things we need to think about when we're designing our probe? How would you design your probe? What's that? The sequence. And what sequence are you going to use? And it's going to be what compared to your gene or your sequence of interest? Complementary. Yeah, it's going to be able to base pair or hybridize. Hybridize simply means being able to base pair to that sequence. So your probe, you want it to be at least 20 to 30 nucleotides long. Sometimes we make them much longer. You want it to recognize one specific sequence so that if it's there, it will base pair. And then what's the other feature we need in our probe? Yeah, so fluorescence or radioactivity. You need a way to be able to detect, did the probe actually hybridize? And so then the final step is you detect. If there was a lot of protected DNA, you're going to see a big glowing band because a lot of probe is going to be able to stick to that DNA. The DNA got all chewed up. There's nothing for the probe to stick to. It gets washed away, and you'll see blank or a very faint band. Any questions about a DNase 1 protection assay? All right, let's do a clicker question on it. So here are the results of a DNase 1 protection assay of the chromatin state of 3 different genes, gene A, gene B, gene C. Which of the following statements are supported by these data? So in each lane, I've either added no DNase, my control, right, or I've added DNase, the same amount to each one. Ten more seconds. All right, put your final answers in. All right, we have a lot of people liking answer D, and then a few votes for most of the other ones. Who wants to take us through this and explain their answer? Someone over here. Yes, back there. I'm sorry, what did you say? You like D, like with the rest of the class? Right, yeah. So D, gene B is likely to be more active than A or C. Why did you choose that answer? Yeah, so we said that when our chromatin is more open, that tends to be associated with an active gene, meaning polymerase can get in there and transcribe it. So it wouldn't be very protected, right? If you're not protected, you're going to get a lot of degradation. Are you going to get much probe binding to it? No. So what you're looking at here, so these bands, what you're visualizing, what you're detecting is the probe. The only way you see the probe is if it had DNA to hybridize to base pair to. And so if we look in our gene B when we've added DNase, we have a very faint band compared to what we're getting for gene A or C. And so when it says gene B is likely to be more active, it looks like gene B was less protected, more open, and they're more likely to be active compared to our other two options here. So D is indeed the correct answer. Does everyone understand that? All right, yes? So we're going to, so that actually relates to, I think, A here. So let's go through each one of these and see why the other ones are not correct. And if that doesn't answer your question, maybe the next slides also will. So, but did you have a question first? So it doesn't have to be completely disappeared, right? But if we're doing in comparison to these two, This was much less protected and therefore much more open than compared to our other two right there. All right, so let's look at these other options here. So A says gene C is smaller than A or B. Okay, so some of you may have looked here and we said that bands run through the gel based on size. If they're smaller, do they run closer to the bottom or closer to the top? Bottom. All right, I see my bands for gene C running closer to the bottom. So why is A not correct? Yes? Nailed it, exactly. So we chose the restriction enzymes to use to cut out a region in these genes that we are interested in analyzing. It doesn't have anything to do with the actual size of these genes. The size of the band is based on the restriction enzymes we chose, which could have just been part of the gene, the whole gene. Some genes are millions of base pairs long, so you're only going to choose a small fraction of that to be able to run into the gel. Does that make sense to people now? If it doesn't, we're going to go on to a slide after this that might make it even more clear. Let's move on to the next options, though. So B says the chromatin associated with gene A is more euchromatic than the chromatin associated with B or C. What does euchromatic mean? Open, right? So euchromatic is when it's the open conformation. Is that what we're seeing with gene A? No. We got actually like completely protected here compared to when we had no DNase to when we added DNase. There's no difference here. So if we're comparing this to our other two options, we get a lot more probe binding here than we did to these. And so if I had changed it to say more heterochromatic than the chromatin associated with B or C, would that have been correct? Yes. So heterochromatic means dense, very condensed, it would be protected. Euchromatic is open, unprotected. Band would be very light or non-existent. C said the chromatin associated with gene B is likely to have more H3K9 methyl modifications than the chromatin associated with A or C. What is one word I could have changed in here and it would have been true? Change methyl to acetyl, right? Because we said gene B here must have been pretty open and pretty not protected because we almost have no probe binding here. Acetylation at that histone 3, lysine 9 is associated with the more open, looser, less protected. And then, of course, it wasn't E because answer D was the correct one. Does anyone have any questions about this? All right, let me just go through a little bit more here to make sure that we all understand why those different sizes of the gene aren't necessarily what we were able to conclude from this type of experiment. So let's say here's our gene that we are interested in, and we know that here's a variety of different restriction enzyme choices we have. And so I'm asking you, would all of these restriction enzymes be good choices to use in a DNase 1 protection assay using this indicated probe, and then explain your answer. All right, so I've designed a probe. Oh no, I put it in the promoter. Is that a problem? Sure. Yeah.
You need to go like between the 5 and 3 primers.
Do I?
So it can be correct.
So in my southern blot, what am I detecting? What is my probe going to bind to? DNA. So this, we are looking only at DNA. So it doesn't matter where in the gene I design my probe, because all these parts will be in the DNA, right? If we were doing a northern blot, which we'll talk about in a few weeks, and looking at the RNA product, I wouldn't detect anything if I put my probe in the promoter. So maybe that's what you were thinking of. But for DNA, we can put our probe in any part we're interested in, right? So it's fine that it's in the promoter, but let's look about our restriction enzyme choices. So EchoR1 is a restriction enzyme. It would cut here. Let's say that length between there is 5,000 nucleotides. Would that work for my southern blot experiment? Yeah, that would be fine. It would cut out this fragment. 5,000 would run into the gel just fine. the probe is within that fragment, I would be able to detect it. So EchoR1 is a good choice. What about BAMH1, the way I've drawn it here? So BAMH1 is here and here. What's the problem here? Mm-hmm. It's what?
It is on an exon.
So it doesn't really matter that it's in an exon, but if I cut out this fragment and use this probe, am I going to detect that fragment? No. If I've designed my probe to be here, maybe there's another BAMH1 site somewhere in the genome, but it could be 50,000 nucleotides away. That's not going to run into my gel. So if I want to detect this fragment, all I'd need to do then is design my probe somewhere else. Make sure it is within that fragment. You can put it in intron. Intron's fine, right? We're looking at DNA here. Does that make sense to everyone? And then you also see how this would be a different size fragment than if I use EchoR1, but I'm still looking at the same gene. And so the size of those fragments on our agarose gel is just defined by where we choose our restriction enzymes to sit. Question?
So the probe has to be between two of the same gene sequences for it to be work, or?
So if you want to ask for this gene, is it in condensed or open chromatin, and I want to make sure that the fragment of DNA is small enough that it'll run into the gel, you choose to cut it out with, in this case, these restriction enzymes, and just making sure your probe is between those two sites. Could I have this probe at this position if I used EchoR1? Yeah, absolutely. It is also between the EchoR1s. Does that make sense? Yeah. What about BFA1? So I have a site here, 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 here. Is that a good choice? No. Some restriction enzymes cut very, very frequently. You're just going to get little chops of DNA. That is not going to give you a nice sized band. And the way I've drawn it here, it would cut right in the middle of our probe site. So that would not be a good choice. What about XHO1? So I have one site here. Not the way it's drawn here, because we have no idea. Maybe the next XOR1 site is here, or maybe, again, it's 100,000 nucleotides away. That is not going to give us a good fragment that we can predict where it should run in the gel and design a probe to run in between those two sites. Question?
How would you always use
Because these sequences are like randomly throughout, not randomly, but there's different sequences throughout the genome. Some genes don't have a convenient EchoR1 site that would cut out what you want. Luckily, there's hundreds of different restriction enzymes that all recognize different sequences. So once you know the genome sequence, it's very easy to predict or to design which sites you want to use. Any other questions? Yes?
At what point does the fragment become too long to work with?
So it depends on what percentage gel you're running. In general, we like it to be between 500 and, let's say, 10,000. So you have a lot of flexibility in designing this. All right, and then the last thing we talked about in class are some elements or sequences on a chromosome that are needed for it to be replicated and inherited. What's the name of one of those sequences or elements? Telomeres. What do telomeres do? Yes, they protect the ending of the DNA or of the chromosomes, and in fact, at the end of class today, we'll talk about how telomeres are synthesized and how they actually have this protective role. So we have telomeres. What else do we need? Yes? Centromeres. And what do centromeres help with? Yeah, equal cell division so that the newly replicated DNA can be divided into the newly divided daughter cells. And then what's the last thing we need? If we're going to copy the DNA, we need origins of replication, exactly. And we're going to talk about DNA replication today and how those origins are used. All right, so based on that understanding, let's do another clicker question. So, 3 different HACs. HACs, what are HACs? What does that stand for? Yes? A human artificial chromosome. Remember we said if we know those elements are needed, we can design a human artificial chromosome. All right, so we made 3 different ones. We introduced them to human culture cells and allowed those cells to divide once. Based on these results, which of the following are possible interpretations of the different HACs that we made? Thank you. I'm so glad you're here. Ten more seconds. All right, put your final answers in. All right, we have another majority, this time for B. Who wants to explain their reasoning for this question? Yes? Yeah, so she said B, which a lot of people also chose. So HAC2 is missing telomeres. And we said that HAC2 is now a different size in many of the cells. If you don't have telomeres, remember they're protective. So we'll learn today, 1, you can't solve the end replication problem, so you're going to get shortening. And then also, if you don't have telomeres, those linear ends are going to be very vulnerable to nucleases, and you're going to get chewing up. Sometimes you can even get 2 chromosomes coming together, sticking together if you don't have telomeres, which would make you twice the size or bigger. So yeah, that makes sense. If you don't have telomeres, you would end up with your HAC being different sizes. Let's go through the rest of these, though. And so what do we think is going on with HAC1? I tell you, HAC1 is only present in 50% of the cells. What do you think it's missing? Could be a centromere. But if you don't have a centromere and you've replicated, you end up with 2 in one cell, right? Well, what would you be missing if? Yes? An origin of replication, right? So if it could not be copied or replicated when the cell divided, there's only 1 copy. So it's going to end up in one of the daughter cells. And so HAC1's probably missing an origin of replication. What about HAC3? HAC3 is absent from some cells and present as 2 copies in other cells. What are we missing there? This centromere, exactly. We're not getting equal division into each. So A is not correct. HAC1 expresses a line transposon. That doesn't really have anything to do with this. We could probably make up some complicated way that having a line 1 does something to interfere with replication maybe, but no, A is not as good an answer as the 2 or a B. And so we already said HAC2 is missing telomeres. That makes sense. We get different sizes. HAC3 is missing an origin of transcription. What's wrong here? Well, we already said HAC3 is missing a centromere. Well, what's an origin of transcription? No, it's a wrong term, right? We're talking about origins of replication. And we said HAC1 is missing its origin of replication. So what I'm trying to train you here is to also read very carefully. This will be very important for when you're taking the test. If there's a single word change, could make this statement wrong. And then finally, D says if the cells are allowed to divide 1,000 more times, HAC2 will remain present in 100% of the cells. We already identified what our problem with HAC2 is, right? So what's our problem? We don't have telomeres. Are you going to stay stable, and are those HACs going to even exist? No, they're just going to get degraded, and so many of them will not be present in the cells. Any questions about this? Yes?
What's the difference in function between the centromere and the origin of replication?
Okay, so what did we say you need a centromere for? It's after you already have replicated, which you need an origin of replication for. So that means you've copied your DNA, you have the 2 sister chromatids, and now the cell needs to divide, and it needs to divide equally into the 2 daughters. That's the role of the centromere. So origin of replication, as we'll learn when we talk about replication today, is where you start unwinding the DNA to actually do replication and copy the DNA. Once you have the copied DNA chromosome, you need to divide it equally into the daughter cells. Does that make sense?
Yeah.
Okay. All right, so indeed, that's what we're going to talk about today is our first major molecular mechanism, and that is DNA being copied into more DNA. And what is that process called? Replication, right? And so here I have a little cartoon, some scientists, they are reading the human genome, and she's like, wow, that's so exciting. Why is that exciting? Because these 4 simple nucleotides encode who we are, what we are largely, right? It's the hereditary material that gets passed on. And so if you think about that, replication is so important, right? To make more of a living object, you need to replicate the DNA. And it needs to be done precisely and correctly and not incorporate a lot of errors. So we will talk about the basic mechanism of DNA replication today, and we'll talk about what we mean by replication being semiconservative. We'll see that DNA synthesis, DNA polymerases always require a primer in order to continue synthesis. We'll talk about what we mean by leading and lagging strand synthesis, the main replication factors or proteins that carry out this process. We'll see that synthesis is bidirectional to take advantage of that unwinding DNA. And then finally, we'll talk about how telomeres are synthesized and their protective roles. So here's a very famous quotation. It was said, "It has not escaped our notice that the specific base pairing we have proposed immediately suggests a possible copying mechanism for the genetic material." Who said that, and what does it mean? Anyone know? If you don't know who said it, that's okay, but what does it mean? If you know about base pairing, what do you know about copying? Yes?
You can always make the copy of the first copy of the strand you already have.
Yeah, exactly. If you know your base pairing rules, you always know what the new sequence is going to be. So if you have a C in the parent strand, the new copy is always going to have a G across from it, and so far. So this was actually at the end of Watson and Crick's famous paper on DNA structure. All right, and this is the point. So parent DNA strands are going to serve as templates. So the template is what gets copied. So they're going to serve as the templates for the new complementary, what we call the daughter strands. The newly synthesized DNA is referred to as the daughter strand. And this will involve the original double-stranded DNA being unwound. What's the general name of the enzymes that unwind DNA? Helicases, right. So break those hydrogen bonds, peel those strands apart so that they can be copied. And they are copied just using our base pairing rules. And then the final product takes us from one molecule of double-stranded DNA to two molecules of double-stranded DNA. And the result could look like this, could look like one old parent strand paired to one new daughter strand. Or we could have, after the parent strands are copied, them going back together and the two daughter strands coming together. The top illustration or the bottom illustration, what actually happens? Yes, and do you remember what that's called? Yes, semi-conservative. And the reason why is conservative means to stay the same, right? And so if the parent strands came back, stayed the same, came together, that's what we mean by conservative. But we know that actually the result is semi-conservative. Semi, you keep one old parent, but then you pair it with the new daughter strand that was copied from it. And the way that we know that this is the mechanism of DNA replication came from what is considered by many to be one of the most elegant experiments in molecular biology by Meselson-Stahl. And we unfortunately don't have time to go through that experiment in class, but I have posted this on the class website, and many of you might be interested in reading that article and how they designed an experiment to determine if replication ended up with the semi-conservative mechanism or the conservative mechanism. All right, so thinking about DNA replication in general, there's sort of 4 different types of things that we need. What's one of the things that we need in order to do DNA replication? Yes? We need proteins to do the work, yes, and we'll talk about some specific types of proteins. We've already mentioned helicase several times, but there's a number of proteins involved. Primers. So I'd mentioned before that DNA replication is going to need a primer, and so we'll see how that is synthesized and where we get this primer from. Yes? Yeah, so enzymes are a type of protein, so same sort of general category. So we're going to need enzymes and proteins. We're going to need a primer. What kind of nucleotides to make DNA? Deoxynucleotide triphosphate, so dNTPs. All right, so we have everything except for what's going to get copied. And what do we call that, the strand that gets copied? The template, exactly. So we're going to need the template. This tells us what sequence to synthesize. We're going to need a primer. And so the reason why we need a primer is because DNA polymerases are not able to start synthesis de novo. And what we mean by de novo is it can't just start putting dNTPs down and linking them together. There always needs to be a pre-existing 3' hydroxyl. And then once you have a 3' hydroxyl, then the DNA polymerase, which is a type of protein, can add on to that 3' hydroxyl using deoxynucleotide triphosphates. So these are the 4 general constituents we're going to need for DNA synthesis. Now, here's another way to look at this. So here is our DNA template strand. It's going 5' to 3' in this direction. Therefore, synthesis must be 5' to 3' in this direction. DNA is always going to be anti-complementary. And so we have the growing chain here. Here is our 3' hydroxyl. This would be the primer to which a DNA polymerase can add on to because there's a 3' hydroxyl. What is the full name of the nucleotide that's going to be added on to next? So what's going to get added onto? What? It's going to be a thymine. And what kind of base? Or what kind of... Are we making DNA or RNA? DNA. So it's going to be deoxynucleotide thymine. And is it going to be a monophosphate, a diphosphate, or a triphosphate coming in? It is a triphosphate, exactly. And so what happens is that incoming dNTP, so N means any nucleotide. In this case, it's going to be this thymine because it's going to base pair to the adenine. We have the 3' hydroxyl that's going to attack the alpha phosphate of this triphosphate nucleotide. And then this will be the leaving group. And what kind of bond do we form now? What do we call that? A phosphodiester bond, exactly. So we're forming, so this is a phosphodiester bond, this is a phosphodiester bond, and here's going to be the new phosphodiester bond that's formed. All right, so let's look at these replication activities that we need. So here we have our template, the 2 blue strands, and in order for them to be copied, we need to unwind them. And the enzyme that does that unwinding in general is called a helicase. It's going to peel away those hydrogen bonds and so that we can see the single strand and start synthesis. Now, as that helicase is unwinding that double-stranded DNA, the double strand in front of it can get stress put on it, and so there's an activity called topoisomerase that will relieve that stress ahead of the helicase that's unwinding the DNA. We said we're also, before we can start any DNA synthesis, we need to put down a primer. And the activity that puts down the primer is an enzyme called primase. Can anyone guess what kind of enzyme primase is or what it is laying down here? Can't be a DNA polymerase because it's starting synthesis de novo. So what kind of polymerase do you think it is? Yes? It's an RNA polymerase. Primase is a type of RNA polymerase. RNA polymerases can start synthesis de novo. They don't need a pre-existing primer. They can just start linking nucleotides together in order to make a short little RNA primer. That RNA primer is going to have a 3' hydroxyl on it. And then your DNA polymerases can start adding on to that 3' hydroxyl of the primer. And in this direction, synthesis 5' to 3', the polymerase can continue synthesizing just following the helicase along. But we have this whole other strand here that's been unwound. Is that going to also be replicated at the same time? Yes, it is. So your cell is very efficient. It's unwound all this DNA. It wants to copy both sides of it, right? And so this is what we have, this term lagging strand synthesis comes into play. So first of all, what we have looked at first down below here, where synthesis is going in the direction that the helicase is unwinding, we call this leading strand synthesis. Like any DNA synthesis, it had to start with a primer. Primase had to have sat down an RNA primer, but then the DNA polymerase can hook onto that 3' hydroxyl and can just continue synthesis. So this continuous synthesis we call leading strand synthesis. Now it's a little trickier for the other side because we also still need to be synthesizing DNA so that it's pairing in that antisense, that 5' to 3' conformation. And so in this case, synthesis needs to be going in this direction. If the 5' end of the template is here, then the 3' end of our newly synthesized DNA is here. And so synthesis has to go 5' to 3' in this direction. So what lagging strand synthesis is going to involve them is going to be multiple RNA primers. So every time a little bit of template becomes single-stranded, primase will come in, set down an RNA primer. This gets extended by a DNA polymerase. And this will continue until it sees the RNA primer ahead of it. This actually gets degraded, and then the polymerase that's synthesizing along will fill in that gap. The final phosphodiester bond is then synthesized by, or is then catalyzed by ligase, where these two ends are pulled together. So this is what we mean by lagging strand synthesis. Synthesis is always in the 5' to 3' direction. It's easy for leading strand to do that. 5' to 3' just follows the helicase along. But on the other side, we have to do this discontinuous synthesis that goes along. All right, let's look a little bit more closely at the factors or the proteins, the enzymes we need to do basic DNA replication. So the example from your book is looking at replication of SV40 DNA. What is SV40 DNA? Anyone know? So SV40 is actually a monkey virus, and it is a double-stranded DNA virus that replicates in the nucleus of monkey cells. And it turns out it uses all of the host cell replication machinery except for one factor it brings in itself. Now, this monkey virus DNA is much smaller. It's just a few thousand nucleotides long compared to our chromosomes that are millions of base pairs long. So we learned a lot about how our own chromosomes are replicated, how our own DNA is replicated, by simply studying this much simpler template of double-stranded DNA. So what has to happen for DNA replication to occur is, so what is the very first step? So if you're going to start DNA replication, what's the very first thing you need to do? You need to open up that DNA. You need to expose the template so you have single-stranded DNA so you know which nucleotides to bring in. What is the general name of the activity that's going to unwind or melt or denature or open up that DNA? What unzips your DNA? Helicase. Oh no, I don't have helicase written up here anywhere. Instead I have, this says large T antigen. Large T antigen is the viral helicase. So when scientists first were studying this virus, they just called this protein large T antigen. It turns out that this is the only viral protein that is involved in replication of the SV40 genome, and that's because this is how it tricks the cell into replicating its own DNA. So if I ask you what is the first activity that you need for DNA replication, please say helicase. Don't say large T antigen. That will only be true if we're talking about SV40 DNA. So first thing we need is helicase to start opening up that DNA. All right, we're going to get stress in front of that helicase, so we have topoisomerase to release that stress. As we open up, unwind, break those hydrogen bonds of our DNA, those strands want to come back together. Those bases want to base pair with each other. So we need a protein that binds to the single-stranded DNA. And that's what RPA does. So these little orange balls, these are replication protein A. It is a single-stranded DNA binding protein. It basically just holds the strands apart so that the replication machinery can use them to do DNA synthesis. All right, so we have the strands, we have the template exposed. First thing we need to do is lay down our primer. And we said that we need to make an RNA primer because DNA can't start synthesis all by itself. And the enzyme that does that is nicely called primase. Primase is a type of RNA polymerase. It will lay down 10 to 15 nucleotides of RNA using the template to put our NTPs together. Then there's a DNA polymerase that takes over. It's DNA polymerase alpha. So alpha is the first letter of the Greek alphabet. So you can think about this as the first DNA polymerase that's going to start adding on to your RNA primer. And it's going to add about 20 or so dNTPs onto that initial primer. Then, if we are doing lagging strand synthesis, so lagging strand synthesis involves looping out this sequence here. Once primase and DNA polymerase alpha have laid down that primer, then you can have DNA polymerase delta loaded on to extend that primer further. And it has some helpers to latch it on to the template. RFC is replication factor C. It helps load pCNA, which just means proliferating cellular nuclear antigen. It's not important to memorize that. But what it does do is important. It clamps the polymerase delta onto the template. So this yellow structure here is clamping delta onto the lagging strand template so that it can then continue DNA synthesis. Now, in our discontinuous synthesis, remember we saw there's a lot of little primers that have to be laid down so we can synthesize in the opposite direction of the helicase. And so what has to happen to those RNA primers? Do we want to have them left in our DNA, our newly synthesized DNA? No, they need to get removed. And so there's an enzyme activity called RNase H. RNase degrades RNA, right? The H stands for hybrid. So that primer is going to be a hybrid of the RNA primer hybridized to the DNA template. RNase H comes in, degrades that RNA primer. There's another factor called FEN1 that stands for flap endonuclease. It also helps remove these primers, these sequences that were first laid down by primase and polymerase alpha. The polymerase will then continue synthesizing until it reaches that gap position, and then that final phosphodiester bond is going to be synthesized by DNA ligase. So we were just talking about lagging strand synthesis. What about leading strand? Do we need these same stuff? Do we also need primase? Do we need polymerase alpha for leading strand synthesis? No? How are we going to start DNA synthesis? Always. You need a primer. What makes the primer? Primase. So it's not drawn here because it already started somewhere back here. But also for leading strand, you need to first start by laying down a primer by primase. DNA polymerase alpha is going to extend that. And then the only real difference here is that instead of using DNA polymerase delta, most of leading strand synthesis is done by DNA polymerase epsilon. It also uses this clamp loader and pCNA to hold it to the template, so it can just zip along that template and continue to synthesize, just following along the helicase. All right, so that's a lot, right? So let's review this. So we said the very first step of DNA replication is going to be unwinding that template DNA. And to do that, we need a helicase. The helicase is what's going to break those hydrogen bonds. Once we have single-stranded DNA, we want to keep it single-stranded DNA, and so we have RPA, this replication protein A, that will bind to the unwound parent DNA. Topoisomerase will relieve the stress ahead of the helicase that is unzipping the DNA. And then regardless of whether you're doing leading strand or lagging strand synthesis, you need to lay down a primer. That primer is going to consist of RNA, so you need an RNA polymerase. In this case, it is called primase, to put down about 10 or 20 nucleotides of RNA sequence that matches the DNA template. DNA polymerase alpha then starts adding dNTPs onto that primer. And then if you're leading strand synthesis, DNA polymerase epsilon will take over from alpha once it gets clamped onto the template by RFC and pCNA, and then it's just going to follow helicase, leading synthesis, zipping right along. 5' to 3' synthesis. Lagging strand is a little more complicated because we have to go in the opposite direction, but in general, it's the same idea. You still need a primer. You're still going to have primase, lay down a primer. Polymerase alpha is going to take over, put in some DNA, deoxynucleotide triphosphates. And then polymerase delta is going to be clamped on using RFC and pCNA in order to make those short synthesized strands, adding onto the primers. The primers are going to get removed by RNase H and FEN1. then the polymerase can fill in the gap that was left by removing that primer, and ligase will synthesize that last phosphodiester bond. Any questions about this mechanism? Reality, it's way more complicated than that, but this is the general mechanism that you should understand for how DNA is copied through the process of replication. All right, so we have a number of different types of polymerases that we talked about in this mechanism. Some of these are very high fidelity, others are low fidelity. What do we mean by fidelity? If something has high fidelity, what is it? Not necessarily tightly bound. Yes? Precise accuracy. Yes, exactly. It's going to copy the right nucleotides and make the right sequence. So if I told you that only 2 of our 4 polymerases mentioned up here have high fidelity, which 2 would you want them to be? Yes. Delta and epsilon, exactly. Those are your high fidelity ones. And the reason for that is the sequence that's put down by primase. What happens to that? It goes, right? RNase H takes it out. It goes away. And it turns out that during the removal of that RNA primer, also what got laid down by polymerase alpha, a lot of that gets taken out also by this Fen1 enzyme. So the goal of these first 2 guys, primase and DNA polymerase alpha, is just to quickly get in and put that primer down. It doesn't need to be terribly accurate. It's going to be removed anyways. But your delta and epsilon, you want that to be very accurate, have very high fidelity, because that is what's going to be in the newly synthesized DNA, in that new daughter strand DNA. And so here's a really cool video. Did anyone look at this yet? I think I had the link in the notes. Nobody. Okay. I highly recommend watching this. It's really beautiful to see how this process of both lagging strand, when you have the looped outside, and leading strand synthesis goes in this computational model. All right, let's do a clicker question. So DNA replication in human cells occurs at a rate of about 50 nucleotides per second, pretty fast. But the haploid human genome is greater than 3 billion nucleotides long. So how can fast-growing human cells divide once every 24 hours? Ten more seconds. All right, final answers. All right, wow. We have a lot of majorities happening with our clicker questions, right? So a lot of people liking answer C. Who wants to share their answer in reasoning? Yes. Sure, however you want to do it. Yeah, so the cell cycle is very specific, and you only do DNA synthesis during the S phase. And some cells are never going to divide again and would never do DNA synthesis. So you definitely don't have the genome just constantly being replicated. It's a highly, highly regulated event. Think about how much energy your cell has to use to replicate its DNA. It really wants to only do that when it needs to be and when the signals are there for it. So right. A is not correct. What about B? Yeah, this one's really hard to envision. 2 chromosomes can be replicated by the same polymerase complex at the same time. No, absolutely not. You have one template that's being unwound, and that can be replicated by a polymerase. In fact, we even have 2 different polymerases doing the leading versus lagging strand. So B is not correct. C says there is more than one origin of replication per chromosome. Yeah, so there's thousands and thousands origins of replication scattered throughout your chromosomes. And that's how you're able to copy the whole thing within this very short period of time. So C, in fact, is the correct answer. And then D said not all of the genome needs to be replicated before each cell division. Well, that's a horrible idea, right? Yeah, we definitely don't want that to happen. Your cells would be problematic. All right, so any questions about that? I think most people got that correct or understand it now, hopefully. All right, so let's look at what is actually happening when a template DNA starts to be unwound. What we actually have are 2 helicase complexes that start unwinding the DNA and going in opposite directions. This produces what we call a replication bubble. It is unwinding, producing these template strands from this common, what do we call this? Where does the replication initiate? What is the name of that DNA element or sequence? An origin of replication. So this is a specific sequence that the helicases recognize, bind to, and when they get activated at a specific time in the cell cycle, there's a cascade of kinases that will activate the helicases so that they can start unwinding that DNA. And so replication is regulated by the cell cycle. And probably in your cell biology classes, you've learned many more details of how that is regulated. We're not going to worry about that right now. And then I told you how many replication forks per chromosome are there in eukaryotic cells. There's thousands and thousands, probably on average about 10,000 per chromosome. But now let's look at this compared to how we have been looking at DNA replication. And so what we have simply been looking at is instead of the bubble here, the example I showed you is what we called a replication fork. But the other half of that, this is what is going on there. And so there's a few things to notice here. So when our helicases are moving in opposite directions, it exposes template DNA for synthesis. And we're going to have replication, leading strand synthesis following the helicase in this direction. And up here, we'll have it following the helicase in this direction, which means we'll then also have lagging strand synthesis on either side of this, but opposite sides as well. So when we looked at the replication fork in our example of how leading and lagging strand synthesis occurs, you also have the same thing happening on the other side, but you just have to remember your rules for 5' and 3' base pairing so that you will have both lagging and leading on the same template, but going in opposite directions on whether you're on top versus the bottom. Does that make sense? All right, so let's think about a problem that happens because of this lagging strand synthesis and when we get to the ends of a chromosome. So for leading strand synthesis, we said the polymerase, once you have your DNA polymerase there zipping along, it's going to be epsilon, it's moving, it's just going to follow the template all the way to the very end. Great, we got a copy of DNA here. But lagging strand synthesis, remember, is discontinuous. You have to keep laying down a primer, laying down a primer, laying down a primer. Uh-oh, what's going to happen to our primer at the very end here? Well, the ones in the middle, those get removed. What's the name of the activity that removes the primers? It's an RNA primer, so you need RNase H, and then there's this Fen1 that also helps take out that original primer. And then delta, which is zipping along here, can then copy this template. And then the final bond, that final phosphodiester bond right here, that's sealed by what? Ligase. It's a ligation event, and ligase does it. Well, what's going to happen here? We have nothing upstream. We have no primer to synthesize to fill in this gap. And this is what's called the end replication problem. If when you get to the end of the chromosome, one side is going to have the primer removed, and if you did successive rounds of replication, what would happen to your DNA? It would get shorter and shorter and shorter and be non-existent. So it was very early on hypothesized that there must be some solution to the end replication problem, and of course we now know that that is solved by telomerase. And so the prediction and the identification of telomerase was awarded the Nobel Prize to these three scientists back in 2009. And in fact, Carol Greider was a first-year graduate student when she was the very first person to identify telomerase activity in the laboratory. And she did it using this weird ciliate, tetrahymena. Why was she using tetrahymena extracts to identify telomerase? Well, let's see. Telomerase is this enzyme that we want to make telomeres, to put these protective ends at the ends of our chromosomes. So does anyone have any guesses on what tetrahymena might have a whole lot of, and therefore probably has a lot of telomerase? Chromosomes, exactly. There's about 40,000 linear chromosomes in this tetrahymena at some points during its life cycle. So that's why this weird model organism was chosen. It's like, all right, there's a lot of substrate in here that needs to be maintained, so there's probably a lot of enzyme, and it was a great guess because that's indeed where she first identified telomerase activity. And it turned out telomerase is a fascinating, very special enzyme. It's composed of two components. Like most enzymes, it's composed of protein. So what you're looking at here in green is the protein part of the enzyme. But then it also is composed of another subunit, and that subunit is actually telomerase RNA. So telomerase enzyme has a protein component and an RNA component. Why is that? So telomerase is this enzyme that has an RNA and a protein component. Does anybody know what the function of either of these two parts might be? Yes?
The protein to actually produce the DNA and the RNA portion to actually go off.
So, all right, so the protein has the enzymatic activity. It's going to make the telomeres. The RNA, what is the RNA doing? So you said adds onto it. Do we add on to the RNA of the telomerase enzyme when we're making a telomere? What are telomeres? They're the DNA at the ends of our chromosomes, right? And so telomeres are just this repeat sequence, this buffer sequence that gets added on to the ends of our chromosomes. So let's look and see what this RNA subunit might actually be doing and what kind of enzyme telomerase is. All right, so let's look at the end of our chromosome. So this space, this single-stranded region, that's where the RNA primer got removed, right? So that's how we have this single strand right here. Telomerase, the protein part, is this yellow ball right here. And what is the RNA doing here? Part of it is base pairing to this telomerase, to this telomere sequence. Let's zoom in on this. So I've just zoomed in on that first diagram at the top. So here we have the telomere itself, the end of the chromosome, this repeat sequence. We have the ball, the protein here. And then we have this red sequence here. So telomerase RNA, depending on what organism, is usually hundreds of nucleotides long. But within that telomerase RNA, there is a specific portion shown in bold here that acts as the template. What are templates? What's a general definition of a template? It's what gets copied, right? It has the instructions for what to get copied as. Aha, so the telomerase RNA is going to act as a template. Oh, wait, here we have the end of our chromosome. What do we see there? 3' hydroxyl. What is the primer in this synthesis here? It's the telomere itself. It is that 3' overhang. So it's DNA, that's fine. There's a 3' hydroxyl there. Now I have a template here, so I know what sequence to add. And so this telomerase enzyme is then going to add onto that last nucleotide, to that 3' hydroxyl, using the RNA sequence as a template. So the A is going to specify a T, T, G. And again, you still have to synthesize in a 5' to 3' orientation, but you are adding onto the DNA. So we are using an RNA template to synthesize DNA. What kind of enzyme is this protein? A reverse transcriptase, exactly. So this enzyme telomerase is composed of 2 parts. The RNA serves as the template. It gives a sequence that needs to be copied. The protein is what does the enzymology. It is a reverse transcriptase. It is going to use that 3' hydroxyl at the end of the chromosome, at the end of the telomere sequence, to add new nucleotides onto. Once that has happened, this whole complex translocates, which simply means it slides down and base pairs to the newly synthesized DNA. So here again, we have template sequence. There's going to be a 3' hydroxyl there, right? And so now this template sequence can be used to add more sequence onto the 3' end of that telomere. And so that just keeps happening over and over again until you end up getting this very long single-stranded region. What can happen is, once this region gets long enough, then your normal primase, the part of this is happening during the replication cycle, primase will say, oh, here's some template sequence. I can lay down an RNA primer here, which DNA polymerase takes over immediately after primase. Which Greek letter? DNA polymerase alpha, right. Alpha will start to synthesize, and then we're doing lagging strand synthesis. So then DNA polymerase delta will take over as it's loaded on by RFC and pCNA. And it can go in, and the final phosphodiester bond can be sealed by ligase here. Again, the RNA primer would be removed. There's still going to be single-stranded DNA here, and telomerase could make this much longer. So that is the basic mechanism of how telomerase is able to synthesize long buffer sequence of repeat DNA onto the ends of chromosomes. Any questions about that? All right, let's do a clicker question. So you determine that the chromosome ends of a new species of amoeba contain the following sequence repeated 1,000 times. Based on this information, predict the sequence of the template region of telomerase RNA in this species. 10 more seconds. All right, put your final answers in. Again, we have a majority for C. Why is C the answer? Go for it. Right, so he noticed that I said the telomerase RNA, so U's instead of T's.
So if we look at it at 3' FWC, which is AAU, CCAAU, and flip it around and make it 5'.
Right, so you need to pay attention to the 5' to 3'. It needs to be able to base pair, and we need to have U's instead of T's. Does everybody understand how that base pairs to that? And that would be the template region of the telomerase RNA. The RNA itself would be much longer, but this is the only part that's available to be copied. This is the template region. All right, that's all we'll do for today. We'll finish up the last few slides on telomeres on Thursday.